We'll continue with the morphology of bacteria in this video. We have finished cell wall in the last class. So we are continuing with the next structure, that is capsule. So in the last class I've told you, if this is a bacterial cell, bacterial cell has got cell wall, cell membrane, and inside it will be having a nuclear material, DNA. And some bacteria, in addition to the cell wall, they have an outer layer which is a viscid capsular material outside the cell wall that is called as capsule. And this capsule or this viscid material may be sometimes well demarcated in some bacteria. Only then you call it as capsule. In some others, it will be like a loose slime layer. It will be like a undemarcated slimy layer outside the bacterial cell wall. Then you call it a slime layer. So some bacteria, it will be well demarcated structure. Then you call it as capsule or capsid. If it is a very loose viscid material just lying outside the bacterial cell, you call it a slime layer. And sometimes what happens is different bacteria will be producing the slime layer and all of them will lie together inside the slime layer forming a biofilm. So biofilm is very important in certain infections like dental plaques and all, where bacteria will be lying together inside the slime and communicating with each other by producing certain chemicals. So what is the role of capsule? The most important bacteria which is having capsule is, one is pneumococcus and the other one is Haemophilus influenza. Both of them causes respiratory infection and meningitis. So when a bacteria is having a capsule, here you can see an image. See, the Streptococcus pneumonia has got a capsule. What happened? A phagocyte is trying to engulf the bacteria and is not able to, because capsule acts as a protective layer and it inhibits phagocytosis. So capsule is anti-phagocytic. So that is the most important point regarding capsule. And what is this capsule composed of? It can be either polysaccharide or it can be polypeptide. Most of the bacteria has a polysaccharide capsule. Exception is the first one you have learned in Koch postulates, Bacillus anthracis. Bacillus anthracis has got a polypeptide capsule. All other bacteria has a polysaccharide capsule. So this is an important MCQ. Okay, so how will you demonstrate capsule? You can do it with the help of a negative staining. So we'll see what is negative staining. In negative staining, what happens is, that is what is shown in this figure. We use a stain called India ink. This stain is a acidic stain. So bacterial cytoplasm, bacterial cell wall, capsule and all are negatively charged. So they will repel the stain. The background will take the color. Background will appear black. The organism will repel the stain. So you can see the organism with the surrounding halo in the negative staining. Background will be black and the organism with the capsule will be looking like a halo. So that is how you demonstrate capsule. That's called as negative staining. Why it is called negative staining is, organism is not taking the stain but the background is taking the stain. So just reverse of the normal stains. Okay. And capsule is antigenic. That means it can induce the production of antibodies. And there is another reaction called Quillen reaction. What we do is, we mix the organism with serum to the capsule. I told you capsule is antigenic, it can produce antibodies. So you mix the organism with anti-capsular antibodies. So when these antibodies coat the capsule will appear swollen. When you look under the microscope, you can see that the capsule has become even larger in size. This is because the capsule and anti-capsular antibodies bind together and leads to swelling of the capsule. So this is called as Quillen reaction. You will learn this when you learn pneumococci. So that is how you demonstrate capsule under microscope. Either you can do a simple negative staining or you can do a Quillen reaction. So regarding capsule, what I want you to remember, because it's a 2-mark question which is asked for the previous year questions, university questions. Capsule, it's a viscid material surrounding the cell wall. If it is well demarcated, you call it as capsule. If it's very loose, you call it a slime layer. Remember the examples for capsulated bacteria, and most of them are composed of polysaccharide. Only exception is Bacillus anthracis, which is made up of a polypeptide capsule. And how will you demonstrate capsule? By negative staining and Quillen reaction. And what is the importance of capsule? Please remember this figure, you won't forget. A phagocyte cannot engulf a bacteria when that bacteria has got a capsule. Capsule acts as an anti-phagocytic factor, so it provides virulence to the bacteria. It is a weapon for the bacteria. So that is about capsule. Now the other structure which you have seen in the first image when you learned the bacterial cell in surface, some bacteria has got a long protruding filament from their cell wall. Sometimes it will be present all over the cell. Some will be having just on one side or on both sides. So this is called as flagella. What is the use of flagella? Flagella is for locomotion. Okay, lot of L in flagella now, so it's for locomotion, or it is the organ of locomotion which helps in bacterial motility. So if a bacteria is having flagella, it will be able to move or motile. And what is flagella composed of? It's composed of a protein called as flagellin. It's easy to remember. Flagella, flagellin. And it has got three parts. You can see in this picture, it's a pictorial demonstration. This is called filament. The long protruding thing is called as filament, which is the propeller. And then there is a hook region. So this is the filament region. This is the hook region. Then it has got two rings, outer ring and inner ring. You can see that the outer ring fixes the flagella to the outer membrane. The inner ring fixes the flagella to the cytoplasmic membrane. So these two composed of the outer ring and inner ring is called as the basal body of the flagella. So just remember there are three parts for flagella. The basal body, which is composed of two rings, outer ring and inner ring, which fixes the flagella to the membranes. And outside, extending from the cell, those outer parts are the hook and filament, which helps in moving of bacteria. So that is the parts of flagella. Then flagella also can induce antibody. It is also antigenic, like what you said in capsule. And with respect to the arrangement of flagella, you can classify bacteria. Like if it's having a single flagella, or if it's having multiple flagella at one end, or multiple flagella all over the body of the bacteria, or bipolar flagella. You can classify bacteria based on the arrangement of flagella. Now, how will you demonstrate flagella? You can see whether bacteria is motile. You can just observe under microscope. You put a slide. You see them through the microscope whether the bacteria is moving. That method is called as hanging drop method. So with the help of hanging drop method, you can demonstrate motility of bacteria. That will help you to recognize whether the bacteria is having flagella or not. Now, there are different types of motility based on some bacteria will be like rapid moving bacteria like Vibrio cholerae. It is called as darting motility. And some will be just stately motion. And there are different types of motility. And how you demonstrate, you can also, other than hanging drop, you can use some special stains for flagella. And also you can use electron microscope because flagella is very thin. If you want to visualize flagella, you need to use a special stain or electron microscopy. Okay, so coming to the next structure, which is important question, spore. So after capsule and flagella, cell wall, which we have finished, spore is the other important question from this part, morphology. So spore, as you all know, spore is like a very resistant form. No, bacteria form spore only in the adverse environment so that it won't cause any damage to it. So all bacteria cannot produce spore, only some bacteria can produce spore. So it's a very resistant dormant stage. Dormant stage means in a spore stage there is no replication. Bacteria will be remaining dormant, and it happens only during adverse environments like depletion of nutrients, toxin formation, toxic product accumulation. If it doesn't get sufficient nutrients and if there is presence of toxins or other factors, these bacteria will convert to the spore form. So in bacteria, spore is formed inside the bacteria, so you call it as endospore. And later on, when bacteria comes back to favorable environments, they will germinate. So the spore will germinate and the vegetative bacteria will come out. So that is called spore germination. So you should see what are the layers of spore. So suppose this is a bacteria, there is a DNA, then what happens is this DNA will multiply forming two copies and a septum forms between the two, and the septum will invade into this layer forming a spore. So this part of the mother cell is called as sporangium, and this is the spore region. And in this region, what happens is this septum, it will form a spore coat. So this yellow thing you can see here, that is called the spore coat. So within this is the spore cytoplasm. The innermost layer is the spore cytoplasm, and the outermost layer, which is this septum, has formed a coat over the spore, that is called the spore coat. And between the spore cytoplasm and the spore coat, there is a layer that is called the spore cortex. So the spore coat is made up of calcium and other resistant things which will help the bacteria to resist the adverse environment. At that time, nothing will happen to this bacteria. It is under a safe protection of the spore coat. So the outer layer is spore coat, the inner layer is the spore cytoplasm. So this is the cytoplasmic membrane, and between coat and cytoplasm there is a spore cortex. And some bacteria, in addition to the spore coat, it will be having another layer called as exosporium. So that gives an additional protection. So these are the layers of spore. The outermost one is exosporium, the external one is exosporium. Then it has got a coat. That is the next outer layer called spore coat. And between coat and the spore cytoplasm, there is the cortex. So these are the four layers of spore. You have to draw this. If it is asked for a 5-mark question, you should draw the structure of spore. And what are the bacteria which has got spore? The most important one is Bacillus and Clostridium. And based on the location of spore, in some bacteria, the spore will be like at the end. In some, it will be in the center. In some, it will be just subterminal, just near the end. So like that, it will be a different classification, different types of spore, central spore, subterminal spore, spherical spore. Like different Clostridium species has got different arrangement of the spores. We don't have to by heart all those, but you should know which are the bacteria which is having important spore forming bacteria, that is Bacillus and Clostridium. And both these are having application as sterilization control. You learned this when we learned sterilization. So when a particular process of sterilization can destroy spore, that means it's a very efficient method. So we use these bacteria as controls to check whether the sterilization process is working or not. So that is about spores. So there's a dormant stage which is formed during adverse environment. Bacillus species and Clostridium species has got spores. So how will you destroy spore? That's what I told you. The most important method of destroying spore is autoclaving, and also you can use sporicide and disinfectants. So we learned that in sterilization class. And the last, ribosomes, mesosomes, and intracytoplasmic inclusions. These are the other organelles which bacteria has got. Ribosome for protein synthesis, mesosomes for respiratory enzymes, which is a counterpart of mitochondria in eukaryotes. And intracytoplasmic inclusions are storage granules where bacteria will store some energy for future use. Like in Corynebacterium diphtheria, CD, Corynebacterium diphtheria, you can see certain granules. That granules is called as volutin granules. So that is intracytoplasmic inclusion granules, which is seen in certain bacteria. And what is pleomorphism? What is involution form? With that, we conclude. So, pleomorphism, that is variation in size. Pleomorphic means it is showing different size and different shapes. Certain bacteria uh, can be seen in different size and different shapes at different times. So, that is called the pleomorphism. And the most important bacteria showing pleomorphism is Haemophilus. And what is meant by involution form? They are the swollen, aberrant forms, that is very abnormal forms of bacteria which is seen in aging cultures, that is prolonged long standing cultures. Nutrients will have depleted. In that time, bacteria will form certain aberrant forms and their cell wall won't be formed properly, so that forms are called as involution forms. So they are aberrant abnormal forms of bacteria. So with that, we finished the morphology of bacteria. We have seen cell wall, capsule, flagella, fimbria, spore, all are important questions. So then now we have to move on to staining and microscopy, the different staining methods which we will take in the next video. So with this I conclude. Thank you.